is a polymer of ribonucleotide. Polymer of ribonucleotide held together by 3' 5' phosphodiester bridge. By 3' 5' phosphodiester bridge and complex compound of high molecular weight. It's a complex compound of high molecular weight. The nitrogenous bases in RNA are adenine, guanine, cytosine, and uracil. It's adenine, guanine, cytosine, and uracil, which replace thymine in DNA. We know DNA have thymine bases and RNA have uracil. RNA molecules are single-stranded nucleic acid. It's single-strand nucleic acid that function in cellular protein synthesis. For protein synthesis and replaces DNA as a carrier of genetic code in some viruses. That's replaced as a carrier of genetic code in some viruses. The main function of RNA is carry information of amino acid sequence. RNA is carry information of amino acid sequence from gene to where protein are assembled on ribosome in the cytoplasm. Gene to where protein are assembled on ribosome in the cytoplasm. This is done by messenger RNA. I think it's RNA's function carry the information of amino acid sequence from where gene to where protein are assembled ribosome in the cytoplasm. Who's done this? The messenger RNA carry the information. A single strand of DNA is a blueprint for mRNA, which is transcribed from DNA strand. So, mRNA is transcribed from DNA strand. It's play crucial role in gene expression by acting as an intermediate between the genetic information encoded by DNA and protein. So it's play in genetic expression. RNA play in genetic expression, acting as the intermediate between the genetic information encoded by DNA and protein, acting as intermediate between the genetic information coded. So here are the types of RNA. RNA molecules are produced in the nucleus of our cell. And can also be found in the cytoplasm. Produce nucleus of the cell. First of all, it's produced of the cell and then found in cytoplasm. Produced in the nucleus. Nucleus and cytoplasm is different site. RNA produced in the nucleus and found in cytoplasm. In both prokaryotes and eukaryotes, the three primary types of RNA molecule are messenger RNA, transfer RNA, ribosomal RNA. The cellular composition of them are messenger RNA 5 to 10%, transfer RNA 10 to 20%, and ribosomal RNA 50 to 80%. Beyond the primary role of RNA in protein synthesis, several variety of RNA exists that involve in post-transcriptional modification, DNA replication, and gene regulation. So, RNA's primary role in protein synthesis involve post-transcriptional modification, DNA replication, and gene regulation. Here's the types of mRNA and their function. Messenger RNA, mRNA, transcribe genetic information from gene to ribosome to synthesize protein. It's transfer. Messenger RNA basically transfer genetic information from a gene to ribosome to synthesize protein. RNA basically synthesize protein. Heterogeneous nuclear RNA, hnRNA, serve as precursor for mRNA and others RNA. So it serves as precursor for mRNA, messenger RNA, and the next RNA is transfer RNA. It's transfer amino acid to mRNA for protein biosynthesis. First of all, need messenger RNA for synthesis protein, and messenger RNA need amino acid for protein biosynthesis. So transfer RNA do transfer amino acid, and messenger RNA do transfer genetic information. For what? For protein synthesis. Their next ribosomal RNA, rRNA, provide structural framework for ribosome. Ribosomal RNA provide which structure framework for ribosome. Then next is small nuclear RNA, snRNA, involve mRNA processing. It's involve mRNA processing. When mRNA is processing, small nuclear RNA, snRNA, play a key role in the processing of rRNA. It's play for role rRNA, ribosomal RNA. Small cytoplasmic RNA, scRNA, involve the selection of protein. It involves selection of protein for export. Transfer messenger RNA. The next RNA is transfer messenger RNA, tmRNA. It's mostly present in bacteria. Add short peptide tag to proteins to facilitate the degradation of incorrectly synthesized protein. When the protein is incorrectly synthesized, so it's need to degrade. So there, transfer messenger RNA add short peptide tag